Have you ever really stopped to think about what makes you, well, you? It's a huge question, right? Well, it turns out the answer is written in a secret code tucked away inside almost every single cell in your body. Today, we're going to crack that code. We are diving deep into the incredible world of DNA fingerprinting. Let's get into it.
So, we all look different, act different, we are different. You'd think that means our DNA must be wildly different from person to person, right? I mean, that just makes sense. But what if I told you that's not even close to the truth? The reality is actually way more fascinating.
Get this, you share 99.9% of your DNA with every other human being on the planet. I know, that sounds crazy. The vast majority of our genetic code is exactly the same. So if we're all that similar, where does all our amazing individuality come from? How can science use DNA to tell us apart with pinpoint accuracy?
Right here, this is where the magic happens. Everything that makes you unique, your hair color, your height, your laugh, all of it is packed into that tiny 0.1% of your genome. For scientists, this little fraction is an absolute treasure trove, and it is the key that unlocks the entire science of DNA fingerprinting.
So how do we get from that 0.1% to a one-of-a-kind genetic ID? Well, here's our roadmap. We're going to start with the basis of your genetic identity. Find the unique signatures in your DNA, learn about the special barcode that sets you apart, see how the technique actually works, and then we're going to solve a crime. Let's begin.
Okay, so we're diving into that 0.1% difference, but our genome has over 3 billion letters. Trying to find the important bits in there is like trying to find a needle in a cosmic haystack. So what exactly are scientists looking for? What's the fundamental reason we're all genetically unique?
The whole idea hinges on something called DNA polymorphism. Now I know, that sounds super technical, but stick with me, it's actually pretty simple. Think of it like a typo in a very long book. If that same typo gets passed down through many copies of the book, it becomes a common variation, a polymorphism. These inheritable typos are the building blocks of our individuality. They're what scientists hunt for.
So we're on the hunt for these genetic typos. But again, with a 3 billion letter genome, where in the world do you even start looking? Well, it turns out there are specific neighborhoods in our DNA that are hotspots for these kinds of variations.
The answer is in something called repetitive DNA. Just like the name says, these are parts of our DNA where a short sequence of letters gets repeated over and over and over again. You can almost think of it as a genetic stutter. And because these sections don't really code for critical proteins, they can collect typos and mutations without causing problems. This makes them the perfect place to find those unique signatures we're looking for. So these highly repetitive, super variable regions have a special name. They're called satellite DNA. Scientists can actually separate this stuff from the rest of our DNA based on its density. For a geneticist, finding this satellite DNA is like striking gold, because that is the target for fingerprinting.
Okay, we are getting really close now. We found the right neighborhood, this satellite DNA. But now we need to find the exact address. We have to zoom in on a very specific type of repeat that is the absolute cornerstone of this whole process.
And that brings us to this, the VNTR. It stands for Variable Number of Tandem Repeats. And honestly, the name tells you everything you need to know. The most important words there are variable, number. It's a sequence that's repeated back to back, and the number of times it's repeated changes from person to person. That's the secret sauce. Let's make this super clear with an example. On the left, you've got individual A. Their DNA has a specific sequence repeated 4 times. Now look at individual B on the right. They have the exact same sequence, but it's repeated 6 times. It's this simple difference in the count of repeats that creates a unique genetic barcode for pretty much every single one of us. So, we have this incredible, unique genetic barcode. But how do you actually read it? It's microscopic? Well, that's where the brilliant work of Sir Alec Jeffreys comes in. He developed this groundbreaking technique that takes these invisible variations in our DNA and makes them completely visible.
So here's how it works. First, you get the DNA out of a sample, like blood or hair. Then you use these special molecular scissors called restriction enzymes to chop the DNA up into fragments. The cool part is, these enzymes cut around the VNTRs, leaving them whole. Next, those fragments get put into a gel, and an electric current makes them move. The smaller pieces move farther and faster. After that, you transfer the pattern to a membrane and add a radioactive probe that's designed to stick only to the VNTRs.
Finally, an X-ray film shows where the probes are, and boom, you have a unique pattern of bands. That's your DNA fingerprint.
All right, that was a lot of science, I know, but now it's time for the payoff. Let's see how this all comes together in the real world. We're about to step into a crime scene investigation and watch this powerful tool do its thing. This is where it all comes together. This diagram basically tells the whole story of a crime. On the left, we've got our evidence. DNA from 2 suspects, labeled A and B, and a sample from the crime scene, labeled C. And on the right, that's the verdict, the final DNA fingerprint. Let's take a closer look.
So the question is pretty straightforward. Does the DNA found at the crime scene match either of our suspects? We're just looking for a pattern that lines up perfectly.
Now, you might be wondering why each person has 2 bands in their lane. Well, quick biology refresher. You get 1 chromosome from your mom and 1 from your dad, right? So for each VNTR location, you have 2 versions, or alleles. In this example for individual A, one version has 4 repeats, the other has 8. And that's why you see 2 distinct bands in their fingerprint.
Okay, now let's connect the bands to the repeats. The crime scene sample has 2 fragments, one with 11 repeats and one with 7. The fragment with 11 repeats is bigger and heavier, so it doesn't travel as far down the gel. The one with 7 repeats is smaller and lighter, so it zips farther down. The takeaway is simple. The position of the band tells you the size of the repeat.
And now, the moment of truth. Let's compare the DNA fingerprint from the crime scene with our 2 suspects. You can see immediately the pattern from sample C does not match suspect A. Their bands are in totally different places, but when we look at suspect B, it's a perfect, undeniable band-for-band match. The evidence is clear. We've found our culprit.
So, we've seen how DNA fingerprinting can crack a tough case, but really, its impact stretches way beyond the courtroom. Let's just take a moment to look at the bigger picture and understand why this technology truly matters. So let's quickly recap the main points. It all starts with DNA polymorphism, those tiny variations that make us unique. We find these variations in repetitive bits of DNA called VNTRs. The technique then creates a unique pattern of bands based on the size of those VNTRs, and that pattern is a genetic fingerprint that's unique to you, unless you have an identical twin.
And remember, DNA fingerprinting isn't just for solving crimes. It's used all the time to resolve paternity disputes, since we inherit our VNTRs directly from our parents. It's also a powerful tool for population geneticists who are studying human history and how we've spread across the globe.
We've come such a long way. We can now literally read the biological code that defines who we are. And as this technology gets even more advanced, it raises some pretty big questions, doesn't it? As we get better at reading these genetic barcodes, what other secrets about our identity, our health, and our future are we about to unlock? The answer, just like our DNA, is sure to be complex and full of surprises.